Hello and welcome students. In our previous session we had discussed about transcription. Okay, so all of you know that transcription is the process of the DNA code forming an mRNA. mRNA is nothing but the messenger RNA. Yes or no? So messenger RNA, since it is an RNA, it will consist of what and all nucleotides. It will consist of a sequence of nucleotides consisting of what? Four different nucleotides. What are the four different nucleotides? Uracil, then adenine, guanine and cytosine. So instead of thymine, uracil will be present in the sequence of RNA. Are you understand? So these four nucleotides will be present in a particular order in every particular mRNA. Yes or no? So that is considered as the mRNA sequence. Now, after the process of transcription, what will happen? All of you know that the mRNA will move out of the nucleus and it will proceed towards the ribosome for the process of translation. Okay, so we are coming to a very important discussion over here. In this session, we'll be discussing about the genetic code. So what is this genetic code? In this discussion, we are going to find out what is the relation between the nucleotide sequence in an mRNA and the proteins that are formed. Okay, so all of you know that proteins are complex structures and they are made up of amino acids. Yes or no? So the simplest form of proteins are nothing but amino acids. Amino acids combined in various manners, okay, in different stages, they will form proteins. All of you know that, correct? So there should be some amino acid sequence. Yes or no? And how are these amino acids produced with the help of mRNA? That is what we are going to discuss in this session. Okay. So in every process of translation, mRNA is converted into, the mRNA sequence is read and it is helpful in the formation of what? In the formation of a peptide sequence. The meaning of peptide sequence is a sequence of amino acids. Okay. So with the help of messenger RNA, a sequence of amino acids are produced. So that sequence of amino acids, how did it come from the mRNA sequence? mRNA sequence consists of nucleotides, correct? So how do these nucleotides produce what? The protein sequence or the amino acid sequence. That is what we are going to discuss. The relation between nucleotides and amino acids. Okay, are you able to understand? So basically all of you should know that every living organism is made up of only 20 amino acids. How many amino acids? Only 20. So every living organism has different kinds of proteins. The proteins may be structural proteins, the proteins may be functional proteins like enzymes or hormones. Are you able to understand? So proteins may be useful for different ways. It may be useful in different ways, but basically all proteins are made up of only 20 different types of amino acids. Are you able to understand? So how many amino acids are there? Only 20. So to form these 20 amino acids, nucleotides must be giving the code. Yes or no? So how is that nucleotide code changed into amino acid, 20 amino acids? That is what we are going to discuss. So in after finding out the process of transcription and translation also, scientists were finding it very difficult to relate between nucleotides and amino acids. They knew that the result of translation was amino acids and the input that is given is a messenger RNA, but messenger RNA consisted of nucleotides, whereas the proteins consisted of amino acids, correct? So how to relate between nucleotides and amino acids? So it was finally a physicist called George Gamow who found out the genetic code. Okay, he related between nucleotides and amino acids, right? So total number of bases that we have in the nucleotides, total variety of bases that we have is 4, and the number of amino acids that are to be produced are 20 different types. Yes or no? So the code that is required must consist of 3 nucleotides. That is what George Gamow found. Okay, why 3 nucleotides we'll discuss, and that those 3 nucleotides will form something called as a codon. A codon is a sequence of 3 nucleotides which forms a particular amino acid. Are you able to understand? So he gave the idea of different combinations, permutations and combinations he worked out. Okay, so different permutations and combinations, how can you work out for 4 number of bases? We will take it as 4 power 3. If 3 different nucleotides produce the amino acids, we can take it as 4 power 3. So 4 power 3 gives us the various combinations of these 4 different types of nucleotides in combinations of what? In each combinations of 3. Are you able to understand? So therefore 4 power 3 we get 64 codons. Okay. How many amino acids are there? 20 different types. Okay. But we get 64 codons. So you might think, why don't we take 4 power 2, sir? Two nucleotides together. See, 4 power 2 gives us only 16, and that will not satisfy, it will not be able to produce 20 different types of amino acids. Can you understand? 4 power 4, if you take more than 3, 4 power 4, right? If you take it, we will get 256, which is a huge number. Yes or no? So many different combinations we'll get, and all these combinations is not required at all, right? Only 20 amino acids are there, 256 combinations not required. So therefore it must be 3. Yes or no? The number must be 3. The magic number is 3 for a particular codon. Yes or no? So 4 power 3 produces 64 codons. This was proposed by George Gamow. But George Gamow did not have the proof for that. That you should note down. Okay. So proof was not given by George Gamow. Only the theory was given that a codon will consist of 3 nucleotides. 3 nucleotides correspond to 1 amino acid. Okay. So therefore, let me write it down over here. 3 nucleotides, they will code for 1 amino acid. Very, very important. 3 nucleotides code for 1 amino acid. Okay. Right. Very fine. So finally, it was found that the codon is a triplet. The codon is a triplet. Okay. 3 nucleotides code for 1 amino acid. Okay. So the proof for this was given by Hargobind Khurana. You should remember this name, very important for board exams as well as entrance exams. Okay. So Hargobind Khurana gave the proof for this one for the codon being a triplet, and it was the proof by chemical method. Okay. He carried out this particular process. RNA molecules. He was able to synthesize RNA molecules with defined base combinations. Okay. So he synthesized RNA molecules with defined base combinations, and he was able to carry out translation with that to check what amino acid it produced. Okay. In the same way, Marshall Nirenberg also gave a cell-free system for protein synthesis. Okay. He found out a method called as cell-free system for protein synthesis. That means protein synthesis without cellular mechanism. Okay. So he was able to carry out protein synthesis without cellular mechanism, and he was also able to find out, okay, which combination of nucleotides produced which amino acids. Now you understand? And he used an enzyme, a particular enzyme, Severo-Ochoa enzyme, which is also called as polynucleotide phosphorylase. Okay, so polynucleotide phosphorylase enzyme was used to polymerize RNA with defined sequences. Okay, to polymerize RNA with defined sequences, and in a template independent manner, so that they were able to form an RNA without any DNA template at all. Right? So without the process of transcription, they were able to manufacture or synthesize RNA, and with the help of that, they were able to produce amino acids. They were able to check the amino acid sequences. Are you able to understand? So this is called as the enzymatic synthesis of RNA, not transcription. Transcription is synthesis of RNA from DNA. So this was carried out in the laboratory and is called as the enzymatic synthesis of RNA. Okay. So these scientists, they were able to manufacture specific sequences of RNA, and from that they carried out translation to produce proteins. They carried out translation to produce amino acid sequences actually, and they were able to verify which codons represented which amino acids. Are you understand? So what is our conclusion? Totally 64 codons are produced. Okay, 64 codons are produced, and 64 codons will represent amino acids. Are you able to understand? 64 codons will be responsible for producing amino acids. So you can see the table of the various amino acids, right, and their codons. It is a table from your textbook only, and the related amino acids are given. Which codons produce which amino acids. So from the table we can find out, we can actually give a conclusion about the salient features of the genetic code. I hope you have gone through the table properly, right? So the salient features of genetic code we can derive from the table. Okay, so the genetic code is actually a triplet. The codon is a triplet, very important thing. So 61 codons code for amino acids. You can see from the table, totally 61 codons will code for amino acids, whereas 3 codons, they do not code for anything. Okay, those 3 codons are given in the last point, UAA, UAG and UGA. They do not code for any amino acid. What is the meaning for that? When the translation process comes to that particular codon, any of these 3 codons, the process of translation will stop, and therefore these 3 codons, UAA, UAG and UGA, they act as stop codons. Okay, so 61 codons code for amino acids and 3 codons, they function as stop codons. Are you able to understand? And the second point, very important point, the genetic code is degenerate. What is the meaning of degenerate? The genetic code is degenerate. Meaning is that each codon will not code for a specific amino acid. Many codons can code for a particular amino acid. Okay, you can see from the table itself, for certain amino acids, some 4 codons, they code for the same amino acid. Yes or no? Correct or not? In some cases, some 6 codons also code for the same amino acid. But each codon will be equated to only one amino acid. Many codons, several codons, they can code for the same amino acid. Okay, so as you can relate, 61 codons code for amino acids, but how many amino acids are there? Only 20 amino acids are there. So many codons, they code for the same amino acid. That is why we call the code as degenerate. Okay, so even though the genetic code is very specific meaning what? Each one codon, if you take, it will represent only one amino acid. One codon cannot represent different amino acids, so it is very specific. But the genetic code is also degenerate, but because many codons, many different codons, they can represent the same amino acid. Are you able to understand? Right, very fine. The codon is read in mRNA in a contiguous fashion. What is the meaning of contiguous? Contiguous also means continuous without any gap, and one codon and the next codon will share a common boundary. The meaning of contiguous is they share a common boundary, which means that it is continuous, one after the other. Okay, so each codon will be read one after the other. Okay, there is no gap in between. That is the meaning, right? So codon is read in mRNA in a contiguous fashion. Okay, the code is universal. Very important point. The fourth point gives us that the code is universal. What is the meaning for that? The code is universal says that for every organism, the codon represents the same amino acids. Okay, so for all living organisms, the meaning of the codons would be the same. But there are some exceptions for that. For protozoans, okay, certain codons might change. It might represent some other amino acids, and in the same way, for mitochondrial codons might represent some other amino acids. Are you understand? So therefore, except mitochondrial codons and protozoans, the code is universal for all organisms. It is the same. Are you understand? Right. Now, these are the kind of points that will be asked in board exams. So you should note down. Okay, such scientist names, George Gamow and Hargobind Khorana, Marshall Nirenberg. Okay, the type of enzyme they use, Severo-Ochoa enzyme. Okay, and such exceptions. These are the things that are used in entrance exams. They are quite often asked in entrance exams. They are quite often asked in board exams, especially in what? In multiple choice questions. Okay, so you should be thorough with all these points. Okay, right, very fine. Let us move on to the fifth point. AUG has dual functions. AUG has dual functions. AUG codes for methionine. Okay, it is written as MET, methionine, and it is also an initiator codon. So the codon AUG has dual functions. Okay, all of you should note down. It codes for methionine and it is also an initiator codon. Okay, just opposite of stop codon, right? So always the process of translation will start with methionine. It will start with the initiator codon. Are you understand? Right. And as all of you know, UAA, UAG and UGA are the stop codons. Whenever translation comes to these codons, the process of translation will stop, and what will happen? Amino acids are not produced. Are you understand? Right. So in this particular session, we have discussed about the relation between genetic code and about amino acids. Okay, so the next process that we should discuss is translation. The next process is translation, and we have seen the relation between genetic code, the nucleotide sequences, and amino acids. Yes or no? So nucleotide sequences, if you take that, 3 nucleotides will represent 1 amino acid. 3 nucleotides lead to the formation of 1 amino acid, and the major molecule that plays a very important role in the process of translating this nucleotide sequence, that is the codon sequence, into an amino acid is the transfer RNA. Okay, the tRNA, all of you should know that. So the tRNA is the molecule which translates the code in the nucleotides, that is the triplet codon, into an amino acid. Are you understand? Right. So how transfer RNA plays a role and what is the structure of transfer RNA, we'll be discussing. Okay. So with the help of the genetic code, let us see how to predict the gene sequence from the mRNA sequence. Let us see how to predict the amino acid sequence. Are you understand? Right, very fine. So this is one sample amino acid sequence as given in your textbook. Okay. So what do we have? AUG, UUU, UUC, UUC, UUU. Here we have UUU and UUC. Right. So this is the sequence given, and in your book itself you can see that it is divided into three bases. Yes or no? Three nitrogenous bases it has been divided into. Okay, but in an mRNA, if you see, all this will be in one single line without any gap. Okay, all this will be in a single line continuously without any gap. That is why the genetic code is said to be contiguous. Are you understand? Right. So each of these 3 nucleotides will form a codon. So here AUG is a codon which represents methionine. So after the process of translation, what will happen? Methionine will be formed. The amino acid methionine will be formed. UUU represents a codon for phenylalanine. So therefore, after translation, phenylalanine will be formed. Are you understand? So in the same manner, what do we have? We have phenylalanine, and all these 3 are also phenylalanine. Are you understand? So all these codons, they represent phenylalanine. So therefore, this is the amino acid sequence that we get. During the process of translation, what will happen? All these amino acids will be joined together using peptide bonds, and this can also be called as a peptide. Are you understand? This can be called as a peptide sequence. It will be bonded using peptide bonds. So a sequence of amino acids can be called as a peptide sequence, or we can even call it as a polypeptide. Are you understand? A long sequence of amino acids can be called as a polypeptide. Are you understand? Right, very fine. So this will be formed after the process of translation. So now we are able to know how to read the RNA sequence, mRNA sequence, and find out what amino acids will be formed. Are you understand? Now in your book they have asked one question. Can you do the same process in the reverse manner? Can you read the RNA from the opposite direction, in the opposite direction, and will you be able to get the same sequence of amino acids? Obviously the answer is no. That is what I have done here. You can see this sequence from right to left. Okay, this must be from 5' to 3', yes or no? So I have written it from 3' to 5' direction. Now you can see what all amino acids are formed. CUU will represent leucine. UUU is again phenylalanine. Here again CU, you can see leucine. Leucine is formed. This is phenylalanine, and finally GUA is valine. Are you understand? So this amino acid sequence is completely different from the one that is formed over here, and surely you can get to know the amino acids. The nitrogenous base sequence, that is the mRNA sequence, will always be read from 5' to 3' direction. Okay, so most of the mRNAs, they will have AUG at the beginning, and AUG is the start codon. From there only the process of translation will start. Okay, so from 5' to 3' direction only it should be read, not in the opposite direction, because the opposite direction sequence will represent another peptide sequence. Are you understand? Right. In the same way, let me ask you one more question. Can we take the complementary sequence of this mRNA and find out what is the amino acid sequence? Will it be the same? Surely you can try it. Surely it will not be the same. Are you understand? So the amino acid sequence formed from the complementary of this mRNA sequence will be completely different. Are you understand? So therefore, that also we should not do. Okay. So from the DNA, just only one template strand will be there. Even though the DNA is a double-stranded helix, only one of it will be the template strand, and from that only mRNA will be formed. And from the mRNA, through the process of translation, the peptide sequence will be formed. Okay. About the process of translation and how tRNA plays a role in it, we'll be discussing in our next session. Okay. Now let us see about mutations. Let us discuss about mutations and how it changes the gene sequence. Okay. So best example for mutations affecting the gene sequence is the disease sickle cell anemia, which we had discussed in our previous lesson. Yes or no? Right. So sickle cell anemia is an example for a point mutation. It is an example for a point mutation, or we can even call it as a single nucleotide mutation. We can call it as a single nucleotide mutation because only one nucleotide is affected, right? So in the diagram for that, you can even refer to your textbook. This is the diagram that was given for sickle cell anemia in your previous chapter. So the normal HbA gene, okay, that is the hemoglobin presenting gene, the gene that produces hemoglobin, is represented like this. It has a sequence in between. Okay, here there will be a DNA sequence. It has GAG, and the other complementary strand will be CTC, right? So from this complementary strand, that is, this is the template strand, and this will be the coding strand. From the template strand, the mRNA is formed. Okay, so the mRNA that is formed from the template strand will be GAG. From this diagram itself, you can know this will be the template. This is the coding, right? So from the template strand, what will be formed? GAG will be formed for CTC. The previous and afterwards also there will be other nucleotides. Okay, but this one is this sequence is the one that is undergoing what? Point mutation, right? So from the mRNA sequence, the peptide sequence will be formed. Okay, so in the first position there is a valine, second position histamine, then we have leucine, then threonine, then proline, and in the sixth position, corresponding to this GAG of the mRNA, what amino acid is formed? Glutamic acid is formed. Okay, so sixth position glutamic acid, seventh position is also glutamic acid. The sixth position glutamic acid is replaced by valine. You understand? Sixth position glutamic acid is replaced by valine because of the change in the mRNA sequence. What is being changed? This adenine will change into thymine. You understand? So there is a change in that. So that produces what? GUG, which is nothing but valine. You understand? Yes or no? Right. Very fine. So this is what happens in sickle cell anemia, and because of point mutation in one nucleotide, the whole amino acid is changing. From glutamic acid is changing to valine. You understand? So valine is the amino acid produced in the sixth position, and because of the change in the amino acid, the whole peptide sequence will now represent the sickle cell anemia peptide, and the shape of the hemoglobin itself will change. Shape of RBC itself will change because of just a point mutation. You understand? So even a point mutation is able to affect the outcome in such a manner, right? So mutations can be point mutations or it can be frame shift mutations. In point mutations also we have insertion or deletion mutations. So insertion of a particular nucleotide in a particular position, in what way will it affect the mRNA sequence? If you are going to add any one nucleotide in between, what will happen? The codons will get shifted. You understand? So the actual set of codons that were there, they will get shifted to one side. Okay. In frame shift mutations, what will happen? Either 3 or multiples of 3 we have to take. So insertion or deletion of either 3 nucleotides or multiples of 3 nucleotides are called as frame shift mutations. So if 3 are shifted towards the right or 3 are deleted, then what will happen? One complete amino acid will be either added or it will be deleted, but the rest of the sequence will not be affected. You understand? Since codons are in numbers of 3 nucleotides, what will happen in frame shift mutations? The other amino acid sequences will not be affected, only one amino acid will be affected. One amino acid is either added or deleted. But imagine what happens in point mutations. If a single nucleotide is added, the whole amino acid in the mRNA sequence will be shifted. Okay, let me give you a small example. Now if you have AUG, CUU and UUC, okay. Now if you are going to add one amino acid to this, one nucleotide to this, then what will happen? Let us say it is added in this position, okay. One U we are adding over here, so CUU will get shifted. So when it gets shifted towards the right, U will go to the next sequence, UUU, and this C will remain separately. Yes or no? So when you are going to add one nucleotide, or in the same way, if you delete one nucleotide, the codon sequence itself is affected. Instead of CUU, we have UCU. Instead of UUC, we have UUU. Correct? Are you able to understand? If they are able to represent the same amino acid, then it wouldn't be a problem. But if they represent different amino acids, then surely the whole sequence will be affected. Are you able to understand? The gene itself is changed. In the case of frame shift mutations, it is in multiples of 3. Yes or no? So therefore, the amino acids, the other amino acids will remain as it is, but the gene will not be affected. Otherwise, only one amino acid is added or removed. For example, the same sequence we will take. If 3 nucleotides are added to this sequence, how will it be? After this, let us add 3 nucleotides. Okay, you can see these 3 nucleotides have been added in a new manner, but the remaining sequences, the remaining codons will remain in their original form. They will remain as it is. Only this one is added extra. Therefore, the other sequences will not be affected. This one extra peptide is this one extra amino acid is added in between. Okay, otherwise the peptide sequence will not be affected, right? So these are the 2 types of mutations that usually take place in the gene sequences, and thereby they affect the mRNA, okay, and thereby they will affect the peptide sequence formed also. Okay, so with this we complete this part of the topics, right? So this is the introduction towards the next process, which is translation. We'll be discussing about translation and tRNA in our next session. Okay, so stay tuned. I'll see you in the next session. Thank you.